So actually, I leapt at the chance because I do study bacteria primarily, but viruses keep rearing their heads all the time. So I was really excited to come and see what the latest and greatest is in the field. So we've been dabbling a little bit. So I'm going to tell you two vignettes today, I like that word, about one on viral classification using genomes, and the second one on connecting viruses to their hosts via single-cell viral tagging. And this is the work of Suzhen Lo and Maria Junkova. And so let's begin, if I push the right button. Okay, so I got interested in the classification side because we've been spending a fair chunk of time coming up with a genome taxonomy for bacteria and archaea called GTTB. And this is basically a concatenated protein phylogeny. So for the bacteria, we use 120 conserved markers. And for the archaea, we use 122. They're somewhat overlapping, but not too much. And so what we do with this tree is then we fix up all the polyphyletic groups, and we also use the tree itself to normalize the taxonomic ranks, to take the idea being to make that somewhat objective rather than subjective. So the question was, is this a crazy idea to consider this for viruses? And if it isn't, we can stick it in that corner. We've still got 2 corners free on the website. So why a concatenated protein phylogeny shouldn't work for viruses? First of all, too many missing orthologs. So if you make one of those matrices, they're very sparse. So one thing that we've noticed with the bacterial concatenated protein matrices is that you can stick in quite incomplete bacteria, for instance, from single amplified genomes, down to maybe 20% or even less, and you seem to get robust placement of those genomes within the tree. So that was one indication, but also if you go and look in the literature, people that are studying higher eukaryotes use these very sparse concatenated protein matrices in order to classify their organisms, sometimes missing up to 95% of the data. So that was the first thing. I thought maybe we could get away with that. But then the second one, which is the common knowledge, is that viral genomes are mosaic. And so you don't really want to be concatenating together a bunch of proteins that have all different evolutionary histories, because you'll just get a big mess. But an interesting paper from Graham Hatfull last year gave me some hope that maybe for at least some fraction of the viral world, that may not be the case. So in this paper, they looked at the evolutionary mode of mainly phages, and they, by comparing the rates of change of the nucleotides against the shared genes within the genome, they came to the conclusion that there is a low flux type of virus and a high flux mode virus. And so in green is the low flux, and in blue is the high flux. And so, and one of the inferences from the paper was that this explains why some studies show viruses to be mosaic, and in other studies they look to be quite discrete, so maybe because of 2 different modes. So I figured, well, if you take the sparse matrix and you make a tree, then at least some parts of that tree should represent the vertical history of the virus. So this is not obviously the first endeavor to make a concatenated protein-based viral phylogeny. Here are some examples, but typically these are using just a few proteins, or if they're using a lot of proteins, like the study in the left-hand corner, they're looking at highly related viruses that share a lot of genes in common, have a lot of genes in common. Of course, there are many, many efforts now to use more of the genome in order to classify viruses at a much greater phylogenetic depth, but these all tend to be pairwise comparisons using gene content or using k-mer or other things like this, and you display it in a number of different ways. And the earliest case of this was the phage proteomic tree from Forrest Rower and Rob Edwards, and they used a combination of basically all based on pairwise distances between the viruses. So you can see that as a tree, you can look at it as a correlation matrix, or you can visualize it as a network. So we began by taking RefSea, the prokaryotic viruses we focused on first, and we used VogDB, which is Thomas Rattay's virus orthologous groups as the point of reference, and that's heavily biased towards prokaryotic viruses. And so what we looked at first is the number of markers, and we looked at how conserved they were across this data set. And as you can see, it doesn't take long before you're down to under 10%. So if you look at the particular, oh sorry, hang on a minute, yeah that's right. So that is the percentage of those 2,262 viruses on the left, and you see there are a few markers that are relatively high, but it quickly gets down to 10%. And if we look at that, you can see there's the terminase is the best coverage there, the large subunit, about 55% of that data set. And then you get polymerases and integrases and other structural proteins by and large. And these have been used individually to create trees and infer relationships. Now, on the right-hand side is the average copy number of these genes. And actually, the majority of them are single copy, which is what you need if you're making concatenated protein phylogeny, because you don't want to be comparing paralogs to each other. So from this, we selected the top 77 single copy VOGs as a starting point for making this concatenated protein phylogeny. So this is the workflow. We basically used the HMMs that are provided as part of the VogDB to fish out these markers out of that data set. We aligned them and trimmed the alignment with Trim AI. We concatenated the marker alignments, and then we did a filtering on that, which I'll talk about in a sec. Then we initially inferred the tree with maximum likelihood, fast tree because it scales well, but the size of this data set is such that you can use other maximum likelihood methods for inferring the trees. And then we had a look, used ARB to visualize and see what we'd got. So with the filtering, just a quick word, we first of all did the standard masking of the individual marker genes by on the columns, on the individual positions, and then we concatenated, and then we did a second filtering by the viruses themselves. If they had too little representation across that whole concatenated data set, then they were eliminated. And this was an attempt to reduce the sparseness of the matrix, so a little bit stringent filtering in the first instance. And from this, actually about 80% of those prokaryotic viruses passed this filtering step. So if you're comparing to the best single marker there, which was the 55%, that's a bit of a increase in coverage. So this is the matrix, and it's Sparse-ish. So you can see that there are in the, whoops, pointing to the wrong thing. So if it's in dark orange, that means present, and if it's in yellow, that's absent. And so you can see that it's sparse, but not 95% missing. And you can see that it's actually clustering of the different single copy markers according to the main morphological groupings. There's a bit of signal there. So when you make a tree from that, when you make a tree from that, so we just call it viral concatenated protein 77 marker tree with those 1,803 taxa that passed. So you see that you, as expected for a sparse matrix, you get most of your highly supported nodes with the little black dots occurring closer to the tips, and that you get generally poorer resolution of the branching pattern near the root. So I guess that's no particular surprise. I think others have seen that as well. We've also shown on this tree the inferred evolutionary mode, and you can see there's some grouping according to that. So the prediction then that you might have got more reliable reconstruction of those low gene flux viruses. So we wanted to have a look and see how does this tree compare to other genome-based reconstructions. So we looked at Victor first, which is from Marcus Goka's group. And here it's a pairwise. You take your genome pairs and you do a local blast, and then you take those high-scoring pairs and you filter them and you transform them into a single distance value, and you make you put that together with neighbor joining. I'm not quite sure what they did, but to reconstruct the tree, like an evolutionary distance tree. And they also managed to make pseudo-bootstrapped distances. So they had some support values associated with this tree. And I think they had somewhere in the ballpark of 600 or 700 taxa in the original publication in Bioinformatics last year. So what we did was we went and picked we pruned our two trees to the common set, which was 408 common taxa. And then we looked at, between their pseudo-bootstraps and our maximum likelihood bootstraps, we looked at the congruent and incongruent splits that occurred between the trees. So on the left here, you have the congruent splits. So there are 218 in the tree. And on the right, you have the incongruent splits. So 185 in our tree, 187 in Victor. So obviously the tree is not exactly the same, but very close actually in terms of the total number of splits. And then on the left-hand axis, you have the bootstrap value or the pseudo-bootstrap value. And so from this, we can see that in congruent splits, you generally have higher bootstrap values. And in the incongruent splits, you generally have the lower bootstrap values. And so we could use this actually using a Uden index to pick a cutoff for our maximum likelihood bootstraps that maximize the true positives and negatives and minimize the false ones. And that came in at 76% as being our. So then we had some sort of index that we could use as a threshold for the bootstrap value, which is actually better than you get in the general rule, because nobody's ever quite clear what is a good bootstrap value or not. So we had something to compare against there. Then next we looked at the V-contact clusters, which is from Matt Sullivan's group. So again, this is a pairwise distance, an all-to-all blast. You do that, and then you do your protein clustering with MCL, and then you do use a presence-absence as well, and you generate this network and you project it up as a network and identify your discrete clusters. And then these were called V-contact groups, which were thought to be equivalent to viral species, and which Matt told us yesterday had a very high congruence to the existing taxonomy. So what we did there was we looked at the V-contact groups. So these are them shown as bubbles, and the size of the bubble indicates the number of members within that V-contact group. By the way, when we did the comparison, we had 1,520 taxa that were common to the two methods that we could compare. So this is the size of each of the V-contact viral species groups. And then the color indicates if we had what bootstrap support we had for that group in our tree. And so if they were supported by that greater than 75% value that we calculated before, we called that monophyletic, robustly monophyletic. If it was resolved as a monophyletic group at less than 75%, it was green. And then if it was polyphyletic in our tree but resolved as a monophyletic group, as a monophyletic V-contact group or a cluster in V-contact, then it came out blue. So initially when I looked at this, I was a bit disappointed because it looks like there's a fair amount of blue there, particularly amongst the larger clusters. But if you go and break those down a bit more, it's not quite as bad as it looks. It just means that, like looking at cluster 1 here, 92% of them are stably monophyletic in our tree, and then you have some additional smaller clusters that are themselves stably monophyletic, but as a whole group that they're not resolved as a single group in the tree. So on the opposite side of that, we resolved additional relationships between the V-contact clusters that were stably monophyletic in our tree. So you can see, for instance, here, instances where you get 3 groups, group 13, and I can't read it now because the lines are over it. But anyway, you can see these clusters that were resolved as stable monophyletic groups within the VCP77 tree. So the conclusions for that is that you can incorporate, actually that's about 70% of RefSea prokaryotic viruses into the tree, compared to 55% for terminase. That should be 80%, by the way. So the tree's mostly well resolved near the tips and poorer resolution near the base, as you expect. It's largely congruent with these other methods, and keeping in mind that they are doing different things. We're picking out conserved markers, and the other methods are based on the whole genome. So actually that's very encouraging for the consistency between the methods. And then the reason that I was interested to go down this route in the first place is because it's an explicitly phylogenetic method where the tree indicates evolutionary divergence, and what we've done with the bacterial taxonomy is to use that relative evolutionary divergence to normalize the taxonomic ranks. So that gives us the potential to normalize viral taxonomic ranks beyond the species. Okay, so the second story, and I hope I'm not going over time, is the connection of hosts to their phage. And this is actually work that started in Matt Sullivan's group, and it was published in 2013 and 2014. And it's a nice, simple idea where you you fluorescently label your viruses, and you allow them to absorb to unlabeled host cells. So this is a cartoon of a flow sorting scatterplot. And so what you see is that via particle size, you get the small fluorescently labeled viruses up in the left-hand corner, and you get the non-labeled larger host cells in the bottom right-hand corner. And when you put those together, then you get the tagged host cells turning up in the top right-hand, then you can sort those, and then you can sequence them. And a nice feature in the original papers was that because you're using a known host, you can grow it on heavy nitrogen, I should say, and so that you can separate it by SIP, so that you're really enriching just for the phage DNA. So that makes it nice and clean. So the DNA that you end up sequencing, you're getting rid of most of the host signal. So we wanted to take this to a single cell level so that we could cover more of the viral host space. And so in this approach, and this is the work of Maria, you have anonymous host cells and anonymous viral cells, and then you allow them to individually attach to each other, and then you sort them out into single wells, so you have one tagged cell per well. So in this case, we don't avoid the host genome. In fact, we want that because we don't know what the identity is. And in the ideal circumstances, and this was going in kind of rather naively, is that you get your host phage pairings through the sequencing. So we applied this initially to human feces. We've got some interest in poo in the lab. And so we ended up using a method which used PEG precipitation to isolate a virions that Maria had previously worked that up during her PhD in her former lab, specifically for fecal samples. And so we did some tests of the viral tagging parameters using pure cultures and the coliphage M13, which is a filamentous virus. And so a key feature after you get your virions is the staining. So in this cartoon here, if you don't get rid of the stain that hasn't been incorporated into the phage, you can get non-specific staining of the host cells, and they actually bounce up here. So with one wash, you'll still have some stain getting through, and if you do 3 washes, pre-washes we're calling them, then that gets rid of all of the excess stain, and you get specific binding. Then, hang on one sec. Then the host cell preparation was simply just to wash to remove the unlabeled virions. This isn't as critical though, because unlabeled virions will interfere, but they're not going to result in non-specific gating of untagged cells. And then we played with the virion absorption parameters a bit. We ended up going with a 1 to 1,000 ratio of host to phage. We allowed them to absorb for an hour at 37 degrees. And then the post-wash is also very important. So after you've got your absorption process, we found that, for instance, that if you don't do enough post-washes, and particularly if you've got filamentous viruses, this is a picture showing that the M13 viruses can non-specifically wrap around a non-specific host, and so you also need to do a number of 3 post-washes in order to prevent that happening. Okay, so we got 11 willing subjects in the group to donate their feces, and then we tried the tagging method, And you can see that between individuals, with the optimized protocol, I should say, you had quite a bit of variation between individuals. Between 40 and about 80% of cells, which is quite high, would be tagged by viruses. So this is taking the viruses from the same subject and the host from the same subject and then allowing the assay to occur. Okay, so to make sure we were on the right track here, before we went to the single cell step, we gated 100,000 cells that were putatively viral tagged, and we got 100,000 cells of host cells that should not be tagged, and then we just did a gene-centric analysis of some sequence of those two sets, and what we could see was there was a clear enrichment of phage genes in the tagged fraction. So that told us we're on the right track. So then we moved to the single cell tagging method itself. So we sorted into 384 wells. We used alkaline lysis with a thermal shock, so at minus 80 degrees and then 55 degrees in order to lyse the cells. And then we used a modified RepLG protocol to amplify up the host viral pairs. Then we screened these amplified genomes by qPCR to make sure that the amplification worked, and then subsequently by 16S to check that there was a host cell there. So we found generally that that was a better indication that we had something that was real and not junk DNA. I know there was a talk earlier on about skipping the 16S step and getting pairs that might be, or bacteria that don't have the 16S gene, mismatched to the primer. And then after we did that, we sequenced and we analyzed, which was by a combination of read mapping and de novo assembly. Let's see, okay, there we are. Okay, so that was, we worked out using the controls the right amount of coverage of a reference template and the depth in order to score it as a positive or a negative, which I won't go into because I haven't got time. So, you've got a number of possible scenarios from the data that you get back. So if you use verse sorter and you identify in your assembly 3 contigs that have been putative viruses, it could be that all 3 contigs are coming from the one virus that have attached, and so they just haven't assembled together. It could be that they represent 3 independent viruses that have all absorbed to the surface of the host cell, or they could be coming from prophage within the host cell as well as from the attached virus. And indeed, we saw all of those options. So this was the workflow that we finally went with. So from the stool sample, we separated the bacteria and the viruses, and we created a bacterial reference metagenome for each subject. And then we did de novo analysis, extract the MAGs as a reference bacterial data set and combine that with reference genomes available from NCBI. We also extracted from there a database of prophages from those references that we'd have prophage reference. From the virus side, we did a cDNA step so that we could capture any single-stranded DNA viruses as well as double-stranded. And then after the viral tagging experiment, we had our amplified pairs, and we mapped those reads back onto our reference host to identify which the host was. We did some de novo assembly, and then in a fairly iterative process, we would go back and forth here in order to try and identify viruses and link up viral contigs in order to identify if what the viral host pair was. Okay, so this is an example of some of the output here. So this is so we clustered the viral sequences in the first instance with pretty stringent parameters. I think it's 95% identity over a majority length of the contig over the overlapping part, and so what we saw here was multiple conformations of specific host pairings. So this is Eubacterium hallei with a virus, and so we had multiple conformations from the single cell themselves, but also we had we found them in the metagenome as well. And this is another instance here, Blautia wexleri with one virus, and this tended to be coming from, we saw the multiple conformations coming from particular subjects. So they seem to be subject-specific. And what we noticed while we were doing this process is that the single-cell viral tagging can help you bin metagenomic contigs that belong to the same viral mag. And so this is a summary of the fecal host network that was produced. So in the ellipsoids are the bacterial hosts, and then attached to them are various types of different phages. So ones that we inferred to be virions are the five-sided objects, and ones that were prophages are this shape, and then ones that were indeterminate were, no, sorry, the other way around. Prophage is a square. And then, so you can see that there were multiple phage that were found to be associated with particular species, and a fairly sizable subset of these could be confirmed by CRISPR. So if they're blue, that means that we had independent confirmation by CRISPR, and if they're surrounded by red, there were tRNAs that also linked the viruses back to the hosts. So we had, in total, we had 163 unique virus host links. That means between a viral species and a bacterial species, and across 42 bacteria and 162 viral species. So that means, in fact, we only identified a single instance of a virion that could target two species, in this case two species belonging to the same genus. So very high, no broad range viruses detected with this initial pass. Okay, so the conclusions are that SCVT is a novel approach for actively mapping the phage-host network. So you actively do an experiment to increase the likelihood of the interactions, and of course you can play with the conditions to increase or to get different phage-host pairings. It's complementary to the informatic approaches. Key parameters such as the washes should be optimized on a case-by-case basis if you're looking at different ecosystems, and no broad host range viruses were discovered in this case. So it just remains for me to thank the people that did the work. So in addition to Sue, Jen and Maria, we had a great support team at ACE. Here we are on our retreat. So Donovan, Josh and Pierre helped with the informatics on this, and Chris is a big single-cell single-cell enthusiast. I was almost going to say fanatic, but I meant enthusiast. And then we have Lee Deng and Matt Sullivan, who have been long-time collaborators on the viral tagging side of things. Thanks for your time, and I'm happy to take questions.